Aşılar insanlık tarihindeki önemli buluşlar arasındadır. Tarih boyunca pek çok farklı bulaşıcı hastalığa karşı aşılarla başarılı şekilde mücadele edilmiştir. Yakın zamanda COVID-19 salgınıyla insanlık aşının önemini bir kez daha anlamış oldu. Covid ve yeni viral tehditlere karşı mevcut ve gelecekteki aşılar temalı konuşması için çok değerli bir ismi sizlere tanıtmak istiyorum. Profesör Matti Sallberg, kendisi konferansımız için Stockholm'dan geldi ülkemize. Kendisine tekrardan hoş geldiniz demek istiyorum, teşekkür etmek istiyorum. Diş hekimi olan Matti Sallberg, İsveç'te biyomedikal analiz alanında ilk profesör olmuştur. Covid-19 aşısı araştırması için Karolinska Enstitü'de bir araştırma ekibine liderlik etmektedir. Sallberg, 40'tan fazla ABD patentinde mucit olarak adlandırıldı ve yılın bilim adamı ödülüne layık görülmüştür. Sözü ben Karolinska Enstitü Klinik Mikrobiyoloji Anabilim Dalı Laboratuvar Tıbbı Anabilim Dalı Başkanı Profesör Matti Sallberg'e bırakmak istiyorum alkışlarınızla.
Yes, first of all, thank you for inviting me to this exciting conference. Unfortunately, my Turkish is not as good as it should be. So I'll do this in English, I hope that's okay. Yes, let's see, do we have the, yeah. So I'm going to talk a little bit about the present and future vaccines against COVID. And I mean, this is not the last time we're seeing a new virus. We're going to see again and again and again, unfortunately. So here we have a number of different interesting hosts. We have the horseshoe bat that probably was the major cause for the outbreak of COVID. Or both the SARS-1 that was in 2003 and the SARS-CoV-2 that appeared now recently. And then we have the fruit bat that's currently spreading the Nipah viruses, which is the Hendra, the Nipah, and the Langia. And there's been local outbreaks in Asia. Here in Turkey and the Middle East, Asia, you also have the Crimean Congo hemorrhagic fever, which is carried by a tick that is about that size and can run 200 meters. So it's, again, this has happened in historic times, it's happened now, and it will happen again. So why do we see these pandemics? I think right now why we're seeing them is that we have a globalized world that's colliding basically with the farming society where you have wild animals. So if you look at how this could have happened, and then I'll get back to how it happened, the most likely origin of the SARS-CoV-2 is the bat. And then it went to an animal I hadn't heard of. It's in English called the pangolin. And presumably it went from there to what we don't really know. It could be an unknown host. It could be the civet cat or something else. And then, unfortunately, it jumped to the perfectly normal man. And then you have the spread from human to human to human. And that's when you have the pandemic. So how does these viruses appear? Well, first of all, I think the key thing with a new virus is really this, that you have one cell that's infected by two different viruses, maybe three, and then a third virus comes out. So that's really the key to developing new pandemics, that you have a recombination, you have a melting of at least two different viruses into a new one. Then again, you have a lot of people and you have a lot of caged wild animals or farm animals, and they all come together. And that's presumably what happened in the COVID-19 pandemic. It's been speculated that it could have started in a research lab or something, but there's actually two science papers that came out that really nicely showed that this is the city of Wuhan, and this is the Hunan seafood market. And the early cases all seem to have a relationship close to that area. And the researchers could even pinpoint a specific stall in the Hunan seafood market where they actually sold these wild-caught animals. So I think there's little doubt that this is really the origin of the current pandemic. And why do we get the pandemic? Well, first of all, no one, or at least there was in 2003, 10,000 people or 9,000 people, but rest of the world, no one had been in contact with a SARS-CoV virus. And not only that, so no one was immune. And then you had the fact that the virus was easily transmitted. And the first version that we had wasn't that easy to transmit. But now with the Omicron, I think most of us here have been in contact with it. So if you look here, what can the immune response do and what do we want the vaccines to do here? Well, we have the cell that's being infected by COVID or SARS-CoV-2. It binds to the ACE2 receptor. How can we prevent this from happening? Well, we have antibodies that targets the surface of the virus that can really prevent this infection. But if this infection anyway happens, so you get the cell infected, then you have the T cells or the cell-mediated immunity. And these cells are the ones that saves us from ending up into the hospital and dying. So even though we get infected, the cell-mediated immunity protects us from severe disease. And looking at the vaccine technologies, I know here in Turkey, you've used a lot of the whole virus vaccines, which are very good. They are maybe not as effective as the preventing infection, but they are certainly very effective in preventing severe disease and death. So this is an old technology that we've used for over 100 years that is really good. But then we have the protein-based, where you have the spike protein expressed and mixed with an adjuvant. And that's Novavax and GSK Sanofi are producing those. But then what really changed everything in large parts of the world was the genetic-based vaccines, the ones that actually transferred the gene for the vaccine, and then our own cells produce the vaccine. So we become our own vaccine factories. And that's really a paradigm shift in vaccine technologies. So as you remember, the pandemic was first started late 2019, and then mid-January 2020, the Chinese researcher released the genetic code of the virus. And that was really the starting point for establishing vaccines. And how did it become so? Everybody, including myself, when people would ask, how long will it take until we have a vaccine? Or I'd say, well, maybe 3 years, maybe 4, 5, 10 years. But that proved us all who said that to be wrong, because the design of the vaccine started in January 2020. The phase 1 clinical trial started in March. That's 2 months later. That's completely unbelievable. It's never happened that quickly before. So how come that the vaccine could come so quickly? First of all, we had the technologies, the RNA, the adeno, the whole virus vaccines. Secondly, we knew how to make the vaccine, because we've done research on SARS-CoV viruses since 2003. So we knew how to make the vaccines. That's why it went so quickly. So it's a combination. We knew how to make it and we had the technologies. So what does these genetic vaccines do? Do they make us magnetic, as many say? No, they don't. They actually make us healthier. They protect us from infection. So if you have what you're injected with is either the whole virus vaccine, but in this case, you're injected with a genetic code in the form of RNA, DNA, or DNA in the form of an adenoviral vector. It's injected into our cells. Our cells start producing the vaccine protein. And then we develop antibodies, we develop T cells. And what these do, the antibodies, the one that we get in the lung and in the nasal cavity, they protect cells from becoming infected. And then the CD4 and CD8 plus T cells, even if we get infected, they will help or prevent us from ending up in the hospital or dying of a severe disease. So these technologies all have their different pros and cons, what's good and what's bad. The RNA, it's really a simple technology. You just produce it, you mix it with lipids, and then you can inject it. The RNA is taken up by the cell, and the cell produces vaccine protein. The adenoviral vectors, they're very good because they induce also the cell-mediated immunity in a good way. But they need to go all the way into the nucleus, and then it produces RNA and the protein. The problem is here, each time you use the adenoviral vector, it's going to work a little bit less effective because we get immune responses to the vector. And then you have DNA vaccines, which work pretty much similar to the RNA vaccine, except it has to go inside the nucleus. So vaccine efficiency, that's been a large thing, that it's 95% effective, it's 65% effective or whatnot. So this is, if we look at the early time of the pandemic, and this is the BioNTech vaccine, and soon, less than 1 month after the 2 doses, you had a 90% efficiency against symptomatic disease. That declined. Unless you're very young, it stays up. It declines maybe now down to 40%. But severe disease and death, that remains. And that, I think, is a really important message. Even though people say, oh, the vaccines are not that effective. Yes, they are. They protect us from death and ending up in the hospital. But then again, now we have the Omicron. And the Omicron is a virus that really can kind of escape our host immune response. So if we look here, this is the BA1 and the BA2, the early versions of the Omicron. Previous infection has a 50% protection against symptomatic infection. 2 doses of the vaccine doesn't protect against symptomatic infection. But if you have 2 doses and a previous infection, then you have 50%. If you have 3 doses of the vaccine and a previous infection, then you have 70% protection. But if you look at against severe disease and death, it's 100% protection almost. It's from 70 up to 100%. So despite that the virus tries to change, we still have a very good protection. And the reason why we had that is because the virus can't mutate away from our cell-mediated immune response. So how do we use these different, I mean, it's a fantastic new era where we can give first RNA vaccines, we can give first the protein-based vaccines, we can give first the whole virus vaccines, and then we give a different one. It's called heterologous prime boost. And in many cases, in animal experiments, it's been shown that if you do this, you vaccinate with one technology and then you come back with another, then suddenly 1 + 1 is not 2, it's actually 3. So this is something, a vaccinology has been kind of a boring, field that where nothing happens, and now suddenly we're testing all the different wild things. And what it really shows, if you give, for example here, 2 doses of the J&J's adeno vaccine, you have a rather limited effect. But if you give first the adeno and then an RNA vaccine, suddenly you have a much better effect. So really, by combining different technologies, which we are doing now all over the world, we actually get better responses than just doing one technology. Now, to finish off here, I'm going to talk about 3 vaccines that we ourselves are developing. One of them is targeting chronic hepatitis B and D. Chronic hepatitis B and D, they are the leading cause of severe liver disease, infectious leading cause. So around 300 million are chronically infected by hepatitis B. And as you can see, it's all around the world. And of these, 20 million also carry the hepatitis D. So we're developing a technology that the antivirals that are available today, they block maturation of the virus. They block the later stage of the viral life cycle. So we're targeting the early stage, the entry. And what we can see here, we use a quite interesting mouse model. It's a mouse model based on a human liver. So these mice can actually be repopulated with human cells. And if we try to infect those with hepatitis B or hepatitis D, that works fine. They get chronic infection. But if we give our vaccine, they're completely protected against both these infections. So this is something that we hope to take to clinical trial to try to treat patients with chronic hepatitis B and D. Then in the next, let's see here. Yeah, we have the Crimean Congo hemorrhagic fever. And this is the hyoloma tick. And as you can see here, it's distribution, and mainly here, also in southern Europe and the Middle East. We have the tick that transmits and becomes a hemorrhagic fever, and it can have up to a 40% mortality. So what we've done is to develop a DNA-based vaccine that can actually, when we inject with a special technology called in vivo electroporation that sends an electric pulse so that the DNA is taken up by the cell produces the vaccine, pretty much like the COVID vaccines. We activate T cells and antibodies, and then when we do this in monkeys, non-human primates, we give them a challenge. And as you can see here, this is the virus in blood in a non-vaccinated animal and in the nose of a non-vaccinated animal. And if it's vaccinated, it's completely protected. So this is also something that we hope during next year that we can take to a clinical trial. And then the last example here is a SARS-CoV vaccine, where we are doing something different than everybody else is using the spike-based protein. We're combining that with other parts also from the virus. So we get an immune response that's much broader and hopefully wider protected. And that one we have here just shown that it can protect a mouse that expresses the human receptor. So here, the non-vaccinated mice, they die very early. The vaccinated mice, they could survive this challenge against the beta strain, an immune escape strain. And finally, we've just got approval from the EMA and the ethics to perform a phase 1 clinical trial with, again, a DNA vaccine delivered by in vivo electroporation. So that's going to start now in November of 2022. 2022, of course. So just to summarize, I mean, with the vaccines, we have fantastic new technologies that gives us completely new possibilities. There's new production. I mean, with the RNA and DNA, you can have one single production line for 100 of different vaccines. And that's really an advantage. It's quite expensive. We don't know how to handle it yet, but I think that's the future. So to sum up, are we better equipped for the next pandemic? Absolutely. We have so many more technologies to use, and hopefully we're doing right now the basic research that prevents the next pandemic. And with that, I'm just going to say thank you to the people I've collaborated with, and thank you for listening. Future